Assalamu alaikum. Today we are going to discuss about growth adaptations. The basic principles are an organ is in homeostasis with the physiologic stress placed on it. An increase, decrease or change in stress on an organ can result in growth adaptations. Number 2. Hyperplasia and hypertrophy. An increase in stress leads to an increase in organ size, occurs through an increase in the size, hypertrophy, and or the number, hyperplasia of cells. Hypertrophy involves gene activation, protein synthesis, and production of organelles. Hyperplasia involves the production of new cells from stem cells. Hyperplasia and hypertrophy generally occur together. For example, uterus during pregnancy. Number 1. Permanent tissues, for example, cardiac muscle, skeletal muscle, and nerve, however, cannot make new cells and undergo hypertrophy only. Number 2. For example, cardiac myocytes undergo hypertrophy, not hyperplasia, in response to systemic hypertension. Pathologic hyperplasia, for example endometrial hyperplasia, can progress to dysplasia and eventually cancer. A notable exception is benign prostatic hyperplasia (BPH), which doesn't increase the risk for prostate cancer. Number 3. Atrophy. A decrease in stress, for example decreased hormonal stimulation, disuse or decreased nutrients blood supply, leads to a decrease in organ size. Atrophy occurs through a decrease in size and number of cells. Decrease in cell number occurs through apoptosis. Decrease in cell size occurs through ubiquitin proteasome degradation of the cytoskeleton and autophagy of cellular components. In ubiquitin proteasome degradation, intermediate filaments of the cytoskeleton are tagged with ubiquitin and destroyed by proteasomes. Autophagy of cellular components involves degeneration of autophagic vacuoles. These vacuoles fuse with lysosome whose hydrolytic enzyme break down cellular components. Number 4. Metaplasia. A change in stress on an organ leads to a change in cell type. Most commonly involves change of one type of surface epithelium, squamous, columnar, or urothelial, to another. Metaplastic cells are better able to handle the new stress. Barrett esophagus is the classical example. Esophagus is normally lined by non-keratinizing squamous epithelium suited to handle friction of a food bolus. Number 2. Acid reflux from the stomach causes metaplasia to non-ciliated mucin-producing columnar cells better able to handle the stress of acid. Metaplasia occurs through reprogramming of stem cells which then produce the new cell type. Number 1. Metaplasia is reversible in theory with removal of the driving stressor. For example, treatment of gastroesophageal reflux may reverse Barrett esophagus. Under persistent stress, metaplasia can progress to dysplasia and eventually result in cancer. For example, Barrett esophagus may progress to adenocarcinoma of the esophagus. A notable exception is apocrine metaplasia of breast, which carries no increased risk for cancer. Vitamin A deficiency can also result in metaplasia. Vitamin A is necessary for differentiation of specialized epithelial surfaces, such as the conjunctiva covering the eye. In vitamin A deficiency, the thin squamous lining of the conjunctiva undergo metaplasia into stratified keratinizing squamous epithelium. This change is called keratomalacia. Mesenchymal connective tissues can also undergo metaplasia. A classic example is myositis ossificans, in which muscle tissue changes to bone during healing after trauma. Dysplasia. Disordered cellular growth most often refers to proliferation of precancerous cell. For example, cervical intraepithelial neoplasia represents dysplasia and is a precursor to cervical cancer. Often arises from long-standing pathologic hyperplasia, for example, endometrial hyperplasia or metaplasia, for example, Barrett esophagus. Dysplasia is reversible, in theory, with alleviation of inciting stress. If stress persists, dysplasia progresses to carcinoma, irreversible. Aplasia and hypoplasia. Aplasia is failure of cell production during embryogenesis, for example, unilateral renal agenesis. Hypoplasia is a decrease in cell production during embryogenesis, resulting in a relatively small organ, for example, streak ovary internal syndrome. Thank you.